So, what do you understand by the term pre-hepatic jaundice? It is called as pre-hepatic jaundice here because the problem in the bilirubin metabolism pathway occurs here before it enters the liver. It is the cause due to the excessive destruction of RBC or hemolysis, due to which there is increased concentration of unconjugated bilirubin in our bloodstream. The causes are inherited hemolytic conditions such as sickle cell anemia, thalassemia, hereditary spherocytes, and G6PD deficiency. Whereas in mechanical hemolysis, it occurs due to mismatched blood transfusion and also in malarial parasite cases. As you know, RBC have a lifespan of 120 days, as RBC contains hemoglobin, which is made up of two parts, heme and globin. Heme is a non-protein part and globin is a protein part, which converts into free amino acid and is reutilized in our body. Whereas the heme is made up of a pyridine ring, which consists of 4 parallel rings bound by methionine group with a ferrous ion present in between. Based on the action of heme oxygenase in the presence of oxygen and NADPH, converts it into a linear chain of 4 parallel rings with the elimination of carbon monoxide and free ferrous ion, which is reutilized back in our body. This linear chain of 4 parallel ring is termed as biliverdin, which is a green compound. This biliverdin, on the action of biliverdin reductase in the presence of NADPH, converts into bilirubin, which is a yellow product formed, and this connects with the albumin present in our blood, which is a plasma protein, to form unconjugated bilirubin, which is transported to the liver for its further excretion and metabolism. But in pre-hepatic jaundice, as you know, there is excess production of unconjugated bilirubin, which the liver cannot handle. So now, for a better understanding about pre-hepatic jaundice, I am going to take you on an amazing ride. So welcome everyone to the hemolysis scope group. Here we have a case versus bilirubin versus RBC, and so now let's begin. Disclaimer: this state is just for fun and educational process, so don't mind anything. Now let's first start with the introduction of all of our members.
Good morning everyone, this is Momin and I will be performing the role of liver steam, the liver.
This is Mrunal Jadhav and I will be performing Miss Heme, that's Momin.
My name is Nandini Thakur and I will be playing the role of the child.
Myself Nandini Gavkare and I am playing the role of RBC.
My name is Nandini Thakur and I am playing the role of bilirubin.
Myself Niranjan Adsure, I am going to be presenting about lab finding and clinical finding of pre-hepatic jaundice.
Good morning all, this is Nas Khan and I will be playing the role of sickle cell RBC as Chrissy.
Morning everyone, I am Rajendra and I am going to play the role of steam.
Ordering the code. Today's case is prosecution bilirubin versus defense RBC. So let's start.
Your Honor, मैं आज यहाँ पे खड़ा हूँ क्योंकि मुझे बहुत ज़्यादा मात्रा में बनाया जा रहा है और मैं अनकॉन्जुगेटेड फॉर्म में हूँ, इसलिए यहाँ पे मुझे कोई भी एक्सेप्ट नहीं कर रहा है और ये सब कुछ हो रहा है इस RBC की वजह से।
Miss RBC, would you like to defend yourself?
Yes, Your Honor. हम क्रिमिनल्स नहीं है, हम तो हार्ड वर्किंग सेल्स है जो कि 24 आवर्स सर्कुलेट होते रहते हैं। हमारी लाइफ स्पैन ही 120 डेज की है। हम अपनी चॉइस से बस नहीं होते, हम पे स्क्रीन अटैक करता है और हम खुद ही पैथोलॉजी के विक्टिम्स है और हम इस कोर्ट में साबित करके दिखाएंगे।
As we have seen the stable state of bilirubin for which he accuses RBC, now let's hear from the witnesses brought by RBC to understand their side of story. Miss RBC, please call your witness.
Thank you, Your Honor. So my first witness is Mr. Liverstream.
Your Honor, this is not my fault at all. This is chaos completely. मैं normally bilirubin को conjugate करके liver के पास भेज देता हूँ, इवन कॉन्जुगेट करके बाइल के थ्रू फ्लो कर देता हूँ। बट करेंटली मेरे सेल्स आर ओवरवर्क एंड एग्जॉस्टेड। मैं अपनी कैपेसिटी से 6 टाइम्स ज़्यादा काम कर रहा हूँ।
Thank you, Mr. University. My lord, we heard the story from the liver side.
As you know here, the liver, Mr. Liver is not guilty. It's the excessive amount of unconjugated bilirubin in our body. So now let's hear more about this case from our second witness.
Your Honor, I have my second witness. अगर आपकी इजाज़त हो तो मैं एक बार my second witness.
इजाज़त है।
So my second witness is Miss Siti.
मैं बायोकेमिस्ट्री बुक की कसम खाती हूँ, मैं जो भी कहूँगी सच कहूँगी, सच के सिवा कुछ नहीं कहूँगी। Your Honor, I tried to flow like normal RBC, लेकिन जब ऑक्सीजन कम हुआ मैं हॉरिबल सिकल शेप में बदल गई। I got stuck into vessel and spleen ने मुझे टारगेट किया जैसे मैं डिफेक्टिव हूँ।
Your Honor, जैसे कि मेरे विटनेस ने कहा, जब बॉडी में ऑक्सीजन की कंसंट्रेशन कम हो जाती है तब इनके अंदर भी जो हीमोग्लोबिन प्रेजेंट है वो लॉन्ग डिस्टेंस में प्रेसिपिटेट हो जाते हैं। उसकी वजह से इनका ये सेल्स इलोंगेट हो जाते हैं और ब्लड वेसल्स में स्टक हो जाते हैं। इसकी वजह से जब स्प्लीन इन्हें देखता है तो एज नॉर्मल आरबीसी समझकर इन पे अटैक करता है। मेरी विटनेस तो बहुत इनोसेंट है, इसमें उसकी कोई गलती नहीं है।
मिस्टर स्प्लीन को बुलाया जाए।
मैं बायोकेमिस्ट्री के बुक की कसम खा के कहता हूँ कि जो बोलूँगा सच बोलूँगा, सच कहूँगा कुछ। Your Honor, it's my job. मैं गैमेट सेल को निकाल के उसको बाहर खत्म करता हूँ। बट हाँ, मैं मानता हूँ कि आजकल कुछ दिनों से ये बहुत ज़्यादा हो।
तो witnesses को सुनने के बाद ऐसा लगता है कि this is a case of chronic hemolysis due to single chip dialysis.
As we have seen from the scenario that Mr. Spleen is also not guilty as he is doing his job completely fine by destroying abnormal sickle shape RBC. Now let's hear from something important from hemoglobin regarding this case.
मैं इस बायोकेमिस्ट्री के बुक की कसम खा के कहती हूँ, मैं जो भी कहूँगी सच के सिवा कुछ नहीं। I was in the cell for a minute and boom, I was floating free in the bloodstream. Your Honor, I was forced to do so. When I was in the bloodstream, I was converted into bilirubin. Bilirubin was further converted by enzyme heme oxidase into unconjugated bilirubin. I was forced to do so. It was not my fault.
और ये सब होने के बाद इसे क्लीन कौन करेगा? मैं आई एम द प्रोडक्ट ऑफ हीमोग्लोबिन कैटाबॉलिज्म एंड लिवर कैन हैंडल ओवरलोड।
मेरे सेल्स एग्जॉस्टेड है, वो काम नहीं कर सकते इतना।
As you know here, the excessive rate of hemolysis is the reason for excessive production of unconjugated bilirubin. Now let's hear our final verdict from the judge.
Okay, तो सारे गवाहों को सुनने के बाद ये कोर्ट इस नतीजे पर पहुंचता है कि this is the case of pre-hepatic jaundice which is caused due to excessive hemolysis because of sickle cell anemia. And Mr. University, you are just overworked and hence you are innocent.
So your Honor, on the basis of all this scene, आपको ये लगता है कि इसमें हमारी गलती है?
No, Miss Abhishek, absolutely not. This is neither your fault nor the fault of Mr. University. This is actually because of a genetic condition which is called as sickle cell anemia. So what happens in this condition is there is formation of abnormal hemoglobin because of malnutrition. There is substitution of acidic amino acid glutamic acid to neutral amino acid valine, and the HBB gene for this is located on chromosome number 11, and the change in codon is from GAG to GUG. So both of you, none of you are the actual victims. Both of you are innocent. Okay, so now the court is adjourned.
Thank you, Your Honor.
So basically, conclusion of this case is this is the case of pre-hepatic jaundice which occurs due to the abnormal sickle shape RBC. Now for further lab finding and clinical finding, I would like to call Niranjan Achure.
Now let's us see about laboratory findings of pre-hepatic jaundice. There is increase in unconjugated bilirubin. As there is excessive hemolysis of RBC, as a result of which unconjugated bilirubin increases in blood. Then there is normal level of conjugated bilirubin. As liver function is not affected in pre-hepatic jaundice, as a result of which the conjugated bilirubin level remains normal. Then there is increased reticulocyte count, as the number of RBC decreases in blood, so to compensate that, the reticulocyte count increases. Then there is increase in level of LDH enzyme. LDH enzyme, that is lactate dehydrogenase enzyme, which is found in most of tissue as well as in cells of RBC. This enzyme plays an important role in anaerobic respiration. Thus, due to destruction of RBC, this enzyme is released in blood, thereby increasing in the level of LDH. Then there is decrease of hepatoglobin, as hepatoglobin binds with free hemoglobin and prevents the toxic effects of free hemoglobin, such as oxidation, and which may result in damage of kidney. As a result of which, hepatoglobin binds with excessive hemoglobin, thus the level of hepatoglobin in blood decreases. Then urobilinogen increases. Then there is no bilirubin in urine, as unconjugated bilirubin is water insoluble. Then we can see conditions such as hemoglobinuria, that is presence of excess amount of hemoglobin in urine, and the home test is positive, which indicate positive autoimmune hemolysis. Then now let's just see about clinical features. The clinical features include mild to moderate jaundice, which results in yellowing of sclera and skin, as unconjugated bilirubin being insoluble in water and soluble in fat, as a result of which it gets deposited in sclera and skin. Then there is pallor due to anemia. Then there is splenomegaly, that is, in case of thalassemia, there is enlargement of spleen, as spleen has to overwork, and as a result there is enlargement of spleen. Then there is normal color stool, and urine also shows normal color in case of pre-hepatic jaundice. The patient may have fatigue, weakness, and tachycardia due to anemic condition. Thank you. That was all from our side.